Okay, hello everyone. Why don't we get started? So it's my great pleasure to welcome Professor Daniel Fisher today to the physics colloquium. He is a named professor for applied physics in Stanford, but the name has changed recently, and we both don't know exactly what that name is.
For political correctness, my name was changed. Yeah.
And he also has courtesy appointments. So he's in applied physics, but has courtesy appointments in biology and bioengineering. He received his PhD at Harvard, then moved on to Bell Labs, and later became professor first at Princeton, then Harvard, and now Stanford. Also, 10 years ago, more perhaps. And along the way, he has received a number of recognitions. So he was elected to the American Academy of Arts and Sciences, and the National Academy of Sciences, and he has received the Lars Onsager Prize from the American Physical Society. So in my mind, Daniel's work can be decomposed into two phases. The first one is solidly in condensed matter physics. And in this field, like in many fields of physics, we cherish simplicity and purity, and think of like a perfect crystal as an idealization of a material. And Daniel focused on dirty materials, disordered materials, and showed that distinct and beautiful physics emerges in these disordered complex systems. And that really has helped change the way we think about glasses, slow dynamics, random magnets, interfaces, and more. And then roughly 20 years ago, the second phase started, where he took on arguably the most messy systems that there are, namely living systems and evolving systems. And again, it was kind of striking to see how much structure and interesting dynamics emerges in these systems and can be described using essentially physical approaches. And once you ask the right questions, and that brings me really to the main thing I associate with Daniel, which is asking crucial questions to the field. I think it was really disruptive. That's my experience to some fields of biology, in particular population genetics, the way Daniel entered this field. But also can be disruptive the way Daniel asks questions during a talk. But they are always crucial questions, and in fact, often cut to the core of these problems. And so I learned a lot over the years from these questions, and also being on the receiving end of those questions. And so in that spirit, please ask questions during the talk. I think you will not mind.
Of course not, yeah.
You will not mind, so during the talk as well. And with that, let us all welcome Daniel.
Oh, thank you, Oscar. It's very nice to be here. My first academic position was teaching in the math department at Berkeley in 1973. And then I've talked in the computer science department and multiple times in the chemistry department. But this is actually the first time I've ever talked in the physics department. So it's nice to be here. Even though I guess officially, according to Stanford, I'm an applied physicist now. So I'm going to talk about some work on sort of evolution and ecology. And I will explain this term red queen, which I gather people under a certain age don't all know what it means. But before I start, I want to acknowledge all the people who did all the work. And this is a group of very talented postdocs and graduate students. The four on the top were involved in the theory work that I'm going to talk about, and then the two on the bottom involved in the genomic analysis that I'll use for some of the motivation. So the basic question I want to ask, you've probably by now at least seen in the newspaper that there's a tremendous amount of diversity of bacterial species in your gut, a wide variety of species. However, the variant that you have in your gut and that someone in the same household has in their gut may be a different variant of the same species. You may have a very similar one to somewhere around across the world. And these transmit around relatively rapidly between people and across the world. But there's many, many different substrains. You also find the same thing if you take a little 1 cubic centimeter sample out of a hot springs in Yellowstone that you find within one species. There's a huge amount of diversity within there, which are all directly competing with each other. And the most striking example I'm going to mention motivation is in a planktonic species in the ocean. So these are species, just single cells that just wander, get mixed around by turbulence. And the question is, why doesn't survival of the fittest drive almost all of these extinct, right? They're competing with each other. Some are presumably better than others. Why aren't most driven extinct? So this is the basic sort of motivating question for this. Now, you might argue this is the most boring part of evolution, is the evolution within a species. But, from a physicist's point of view, if we can't understand that, we can't understand much of anything else. But also, the diversity within a species and diversification is what gives rise to the variation, which eventually gives rise to other species and different phyla and so on. So I'm first going to give some motivational data, and then going to really concentrate on what one can gain by thinking of biology as being very high dimensional and the questions that come from that, and then look at some very simple models of the combined dynamics of the ecology and evolution linked together. And part of the goal of this, and is really one of the goals of theory in biology, is which things shouldn't one be so surprised by, right? So if you find some phenomena which seems amazing, and then you find really simple models that can exhibit that, and maybe especially if it's in a reasonably robust way, the models may have nothing to do with the actual process, but the fact that it can occur simply means maybe you shouldn't be so surprised by it. So that's one of the goals here. But now I'm going to ask a quiz. Okay? So 50 years ago, I learned from Freeman Dyson, heard some lectures on Freeman Dyson about human impact on the atmosphere and carbon dioxide in the atmosphere and things. And at the time, the species which dominated the impact on the globe was not humans, but was some other species. In fact, but that other species actually hadn't been discovered yet. It wasn't discovered until into the 80s. Okay? Nobody call out the answer, but how many of you know what that species is? It's still the second most important species as far as impact on the climate. Okay, Oscar is raising a hand. 1 or 2, okay, 3 maybe. Okay, this is pretty bad education, right? That this most crucial species is not known. I didn't know about it then, of course, either until much more recently. So this species is Prochlorococcus. So it's a cyanobacterium. So the cyanobacteria, the ones that do make oxygen, do photosynthesis and make oxygen, they're the chloroplasts inside plants. So that something's only evolved once. There are rather a lot of them, about 10 to the 27th in the tropical oceans. They dominate photosynthesis in the tropical oceans and fix more carbon than all human croplands put together. Okay. One of the surprising, Penny Chisholm discovered them in the 80s. So one of the surprising things about these is how diverse they are. They're very simple. They're very small bacteria, very simple. They don't live on other organisms and things. They just float around. So why you think they're pretty simple? Why should there be much diversity? Okay. So within these, they're rather hard to culture in the lab. And so with recent developments in DNA sequencing, you can take individual cells and you can sequence the individual cells. So you can take a bucket, pull out 100 cells from that bucket, sequence each of them. And it's one bucket, the same species, you'd expect them to be similar. But in fact, that's not what you find. You find they're very different. So this is now plotting. You take 2 of your sample that you've taken. You take 2 random ones from that, or eventually take the whole list. So along the bottom here is all of the ones in the sample, and then all the ones sample again, and then you just compare them. So you look at how far apart they are genetically. So these ones here, now on this scale, these are differing by some of order 20% or so. Nevertheless, they're what's called the same species. It probably really should be called a genus. But you notice sort of structure inside here. And these structures inside here are things which are more closely related when we want to call those species. Now, if you go inside one of these here, you look in one of these expand. This is actually the dominant species by far within this. And then you start seeing it looks sort of pretty uniformly yellow. They're all sort of a few percent apart genetically. But the little tiny clusters in it, you can expand within those, and you see even finer structure there. But on this scale here within the species, they roughly seem that most of them are similar genetic distance apart. And that's roughly the same with people. If you don't not have recent African ancestry, then you take 2 genomes that sort of comparable distance apart to other 2 people's genomes. But these are bacterial things. They're primarily asexual organisms. So the question is, what on earth is sustaining all this diversity? The closest relatives that have been found or have a common ancestor maybe a few hundred years ago. These ones go all the way to tens of millions of years. There's only things on all scales. So the evolution has to have been ongoing, the diversification of these, some variants going extinct and so on, for a very long time, while they're mixing globally on a time scale of tens of years. Okay? There are not many ecological distinctions and so on. So the big question is, how can such extensive diversity evolve and persist when, I say, on decades time scales, they're all competing with each other? Okay. So what are the explanations for diversity? So these are depending on your field you come from, and your biology tends to be very much divided into camps. So even within those fields, there are camps. So the common one, which is that saying they're ecological niches, that each variant somehow has its own ecological niche. Well, if you want to explain a lot of diversity, you have to say there's some micro niches, pico niches there. Well, really, you have to go all the way down to yocto niches. A yocto niche is the inverse of the number of different genomes of Prochlorococcus in the ocean. It's 10 to the minus 24. So obviously that doesn't become a very useful explanation by the time you have to invoke all these detailed differences between them. And the mixing, mixing from the turbulence and so on, and the competition smears out the gradients of resources and other things that might sort of distinguish between niches, and it's very hard to find any such structure in this population of this highlight to Prochlorococcus. The other explanation, which is to say there's nothing interesting ecologically at all, they're all ecologically equivalent. The differences between them, they're in the genome, so they have no biological significance. Everything is controlled by fluctuations in how they move around and births and death fluctuations. Okay? The problem with this explanation is that it's wrong by somewhere more than 15 orders of magnitude if you start putting in numbers. These two processes are much too slow for a very big population like Prochlorococcus. We know that continual evolution, mutations always arise, that can give you rise to a lot of diversity in populations, even without any ecological distinctions. The problem is that the diversification you get from the evolution by itself can't sustain for very long periods of time, and so you can't have these things with common ancestors, it's going from the whole span from hundreds of years to millions of years. These divide once a day. And so if you think of how many generations that is, it's a very large range in generations they would have to survive for. And so we know you can't get this amount of diversity from just the evolution by itself. Okay? It's been said for a long time, and it's still said, that pathogens promote some diversity. Okay? So bacteria attacked by viruses is called phages. But people don't really work through this. Are they specialists or generalists? Is this stable to extinction of the phages, or maybe extinction of the bacteria? And you start looking at models with this, and you find it actually doesn't work very well. So it's not clear that's actually an explanation, but we're going to explore this one in a little bit of depth, at least. The other consequence, being a statistical physicist and liking to wave one's hands, it's a general consequence of biological complexity, whatever one means by that. Okay? But to do that, we really need to have scenarios with simple models, some theory, and hopefully some kind of robust understanding that one has a sense of which kind of features are the crucial ones and maybe which ones don't make so many difference. Okay, so the evolution and the ecology are all coupled together. The evolutionary changes change the organisms change their environment, particularly bacteria that feeds back, and the modified population then is evolving in a slightly different environment caused by its ancestors. Okay? So the crucial thing is that all of this depends on how you interact with the environment, how the interacts affect you, and that's the organismic properties, or called the phenotypes. It's not your genotype. Right? And one of the big problems of biology is going from the genome to what the properties of the organism are, and we only barely know how to do that for single proteins. We don't still know how to do that with individual cells. So most of the information on this fine-scale bacterial diversity is from the genome, so we don't know what it corresponds to. We can't directly measure on that. But when we build any theory on this, one really wants to build a theory from the beginning on the phenotypes, because that's what's going to matter. Okay. So I don't know what complexity is, but I do know something about high dimensions. By any measure, biology is very high dimensional. Okay, what do I mean by that? If I look on the molecular scale, so I look at all the properties on the molecular scale which can affect the organism, particularly affect how it does basic functions that it needs to do to survive and grow and divide. Okay? So the number of such properties in simple bacteria is certainly millions. And so this is a very high-dimensional molecular scale phenotype. You need a lot of numbers to specify this property. If you look at the properties of the organism and how it interacts with its environment, that's a big number, but still a small number. Say all the chemicals in the environment that it cares about, all the ways the chemicals that it can make in the environment, and other things that it can do as well for more complicated organisms. So the dimension that is very large, not as large as the molecular one. The fact that it's not as large is associated with a lot of the redundancy of biology, that there are a huge number of different ways molecularly of doing the same thing at the level of the organism. And that's sort of crucial on how evolution works. It tends not to get emphasized too much. Then there's the space of the environment. And as I say, the simplest is just the number of chemicals in the environment that the organism is affected by and that the organism affects. So things that it can eat particularly. Okay. But then there's an absolutely crucial observation, which is that the complexities of the biology, the ecology, and most especially the evolutionary history means that the difference between any two strains, how they are behave in one environment, which one grows faster, which one dies faster in that environment, how they depend on some changes in the environment, so take this same strain from here to a different environment, that all of those things and the way they couple other biology, everything is conditional. Okay? Meaning that there are all sums, anything like that, the differences are sums of positive and negative contributions. Okay? How do I know that? Well, if something, genetic change, was always beneficial, it would have taken over the population millions of years ago, and you would never see the distinction anymore. So everything you see, all the distinctions between these, when they're all mixed together like this, is either conditionally good or conditionally bad, depending on circumstances. Okay? And that's going to play a crucial role. And I'll come back at the end and make one comment about sort of direct evidence for this. So once you've got sums of positive and negative things, then the first refuge of scoundrelous statistical mechanicians is to say, okay, let's approximate the complexities by randomness. Okay? So what we can then hope is that some of these complexities, sums of positive and negative things, we can at least caricature by randomness and hope that some universality in a loose sense in high dimensions. Okay? And so in the analogy with the stat mech, we really sort of want to think about whether there are some kind of eco-evo phases that involve ecological interactions and the evolution, and there are some phases, and some particular species may be in one phase or the other, but if we can characterize them in those ways, then we can maybe hope that there are some robust features that don't depend upon all of the details. Right? And again, we can get a sense of the kinds of phases that can exist. Okay. So some of the basic questions then with the high-dimensional biology. So initially I'm going to take the simplest conditions of all, completely mixed conditions. So the environment is just uniform and with no temporal variations in the environment. Of course, the organism can cause temporal variations, but none there in the physical and chemical environment initially. Okay. And then we want to ask, can the weak ecological feedback, so this is the changes in the environment caused by the ecology driven by the evolution, can this cause continual evolution even without diversity? So even if I just have one strain that's evolving along, no diversity, what does that do? Then we will want to turn off the evolution and ask just about the ecology on its own and say, can ecology sustain very large diversity within a species without invoking niches? Without saying, oh, geez, this one does this does one this. One has to have some more statistical way to approach that. Then we want to combine them together, and we want to ask, can these ecological communities evolve high diversity, and particularly whether that can happen with small effect mutations rather than big changes. Can this diversification, this ongoing process, diversification and extinctions, continue indefinitely? And then a question which is, I'll say a bit what I mean, how much complexity is enough? Do I have to invoke all the complexities of the biology to get some of these things happening, or is it sort of relatively simple features can already do it? And then the rather crucial question is, are these the right questions? Okay, so I'll come back to that one at the end. Okay, so the first question we're going to ask about the ecological feedback. So I'm just going to have a single strain which is evolving, so there's no diversity there, and ask what will happen, and static environment. Okay. So I can think then of the phenotype, the properties of this organism. So that's some high-dimensional vector, vector X. In the ideal situation where I'm a perfectly fixed environment, right? So the organism is not affecting the environment. It's entirely controlled by the experimentalist. Extremely hard to do and rarely done. In that case, there is the fitness as a function of the phenotype. And I can think of this function as being some landscape. People talk about a fitness landscape. And the fitness is just the difference in fitness is relative difference in growth minus death rate. So if you're more fit than another one in this symbol thing, then you take over on the population. Your population grows, theirs goes off. Okay. So the simplest then is I just got one genotype corresponding to this one phenotype, one value of X, a very low mutation rate, but occasionally a mutation will arise with small effect dx. And then say the selection's fast, it takes over. If it's better, it takes over before another mutation. So what this tells us is that evolution is approximately deterministically uphill in this fitness landscape, deterministically uphill with some stochasticity to it. So what this takes us then is we want to understand things, dynamics in high-dimensional landscapes, and these complexities we're going to model by randomness, and so we're going to look in high-dimensional random landscapes. Okay, so that's a subject which a lot of people in various areas of physics have worked on in recent years, and quite a lot is understood that we can build on, but then we have to add features to it, which are the crucial and most interesting parts. Okay, so as long as the landscape has some structure to it, there are going to be, in addition to maxima and minima, there are going to be lots of saddles, and these saddles are what determines where trajectories go. So here's a saddle, here's different order saddles. You'll go from one trajectory like that. So if you change slightly different, you'll go in a different direction there. So these saddles sort of control things, where things go. Okay? Now to random landscapes which are sort of sufficiently rugged, then there tend to be exponentially many maxima. There are even more saddles, and even more saddles of different orders, of higher order saddles and so on. But of course, what happens is we're doing gradient descent. Why physicists tend to go down landscapes, biologists go up them. They're more optimistic, maybe, except these days. And so we're going uphill. So we know it's going to keep going up, right? It can't suddenly go down and something like that. So it's going to keep going up. And not surprising, you'd expect it to slow down. And it starts going to saddles of lower and lower index, gradually getting towards maxima. But the rather surprising thing is it never really commits to any maximum. It gets near to some maximum, and it's wandering off around saddles, keeps wandering around. In the simple models, wanders over the whole space, getting gradually slower and slower, getting close to sort of borderline stable peaks, where they have a marginal eigenvalue for their stability. Okay. So this is already a little surprising. This is very different than the caricature of what you would get in low-dimensional landscapes, which are full of evolution ecology books have such pictures in them, as well as physics books. But in high dimensions, intrinsically very different. In particular, this keeping to wander, even though it slows down. Okay? Of course, this also means that it's highly sensitive to initial conditions and highly sensitive to perturbations from this as well. And so where it goes, the trajectories will depend upon be very sensitive to other things. Okay, but now, of course, we don't have just this. We don't just have a landscape in one environment. We have to worry about what it looks like in a whole lot of different environments. So we have a whole family of landscapes, of environments, the function of E, say the concentrations of chemicals in the environment. Now, the problem is that fitness, in spite of having two S's, is usually treated as if it's one thing. Right? It's one thing, not a plural. There is a fitness of this organism. And you go, wait, what do you mean by that? Is it on this time scale or on a longer time scale or here? Or what does it mean by that? So I generally tend to be opposed to adding ohms to things and turning things into ohms. But I'm trying to get into the literature, the word fit gnome. So this is, at least it tells you that it's a complicated function. Right? It isn't just one thing. So at a very minimum, it's your growth rate, which is a function of your phenotype, and the function of the environment. Right? Even if there's no spatial gradients or anything else that's complicated. Okay, so we now want to consider these things generally, but of course the interesting part then is that the ecology is going to affect the environment, the organism affect the environment. So the simple situation where we've got one strain, they're all identical, they will then grow until they saturate. They'll eat some of the food and so on, their population will saturate, go to steady state, and they will then affect the environment. It'll be effective whatever physical and chemical constraints you've put in, plus the effects of the ecology. And I say typically with bacteria, that completely dominates what the environment is. It's much more important than what you did to the environment as an experimentalist. And then what we would like to have is we would like to look at this, and we want to have that the evolution is occurring not in a landscape, but it's really in a snowscape. Right? As the evolution occurs, it changes the environment. So it's like you go up a mountain, and you go up a mountain, it's getting steeper, and it's getting snowier and snowier, and you think you're keeping going up. You take a step, but then you slide also. You take another step, you slide there as well, and you think you're keeping going up, but you're actually not going anywhere at all. Okay? So this is the Red Queen evolution. If this can happen, Red Queen is from Alice through the Looking Glass, and one of the main characters in there is the Red Queen, and she's running very, very hard just to stay in place. And if you want to go even faster, if you want to go forwards at all. So this is something where the process looks as if it's going fast, like you're trying to climb up this snowy mountain, but you're not actually getting anywhere. So this is a metaphor, and the question is, can something like that occur? Okay, so the model, the dynamics one puts in is called adaptive dynamics, where you just sort of gradually change the environment. And so the crucial thing here is this mutant with x plus dx. If its fitness in the environment set by x, its parent, is bigger than the original one, then it can take over. It can take over, and it modifies the environment. Okay? That modifying the environment now has changed the landscape. So we now have our dynamics is still simple, but it's now dx dt at constant environment set equal to this. So this is no longer gradient descent. Right? This is dynamics that has curl-like parts if you want. It's no longer gradient descent. Then you can say there's some parameter which defines the magnitude of this feedback, how strong this ecological feedback is. So what will happen here? So here's a saddle, and here's some trajectory going along, and evolves is determined by the saddle. But supposing now we get a bit of evolutionary of ecological feedback. So it's coming towards the saddle, then as it comes towards the saddle moves, and that can make it move enough that goes in a different direction. Okay? So it clearly can affect where it goes. The question is, does it continue to affect where it goes, or is just some initial transient so that it'll occasionally do this? Okay. In simple landscapes, at long times, you've got a few maxima, you'll tend to go to one, and it doesn't matter much. But as soon as you get these rugged landscapes, lots of saddles and things, we already said they were highly sensitive to the feedback, and so maybe it can do something much more interesting, and it does. Okay. So a generic phase for high-dimensional random landscapes, rugged landscapes, is that the environmental feedback, any magnitude of feedback, is enough that it can make it continue to evolve forever. So you gradually, you go up the mountain, then you go along, but then you stop being able to actually go up anymore, even though you think you're stepping forward, and you just keep on going without any further slowing down. Okay. So you can, if you want, you can say how things sort of scale when the feedback is small. This is deterministic dynamics in this model. It's just deterministic dynamics, which is not quite gradient descent. So it's high-dimensional deterministic chaos. This is evolutionary chaos here. Okay. What do I mean that you keep on going without any further slowing down? Well, so a way you can ask is if a mutation comes in, and then the one with that mutation takes over the population. So you can just count by DNA sequencing how often mutations are coming up and taking over. Okay? And this would say that eventually this local fitness flux would go to a constant. The rate at which mutations are coming up in population and taking over doesn't slow down. Okay? So this says in this, at least in sort of abstract models for this, that one can get this to occur, and one can get continual evolution here with only a little bit of the ecological feedback causing from just this one strain which is evolving. Okay. So this is saying we can get a perpetually evolving low diversity phase. Okay. How much complexity is enough for this? Well, you can make a sort of caricature model of the fitness, how it depends on what's going on inside the cell, just in terms of a lot of constraints, how it couples to the environment. You just say there's some optimal direction in the environment given by some h. And then the ecological feedback is just the optimal direction of the environment slightly change as you evolve. Okay, so this is a model which is at least a reasonable caricature. It's got these sort of soft constraints inside the cell. Well, this is enough that for any feedback, it'll continue, the evolution will continue forever. Okay, so we can have ones which are a little bit better caricatures of the biology, and this is a rather robust phase. Okay. So the continuing forever of the evolution of ecological feedback, that's not surprising. The question about the diversity. was what we started with. Okay. So the problem is with this model is if you have a small change dx, it's very hard for the parent and the mutant to coexist because the sort of effective interaction is second order. So you can't really get coexistence easily here unless the evolution takes you in special parts of the landscape. So if the evolution is taking you special parts of the snowscape, then you might be able to get coexistence. Now, of course, with theory, 1's easy, 2 is harder, 3 is ridiculous, so you do many. Okay, so we'd like to understand this, and we'd like to ask about things that can happen with many strains. Okay? So the basic question, the high-dimensional biology, is whether we can get the ecology to sustain a lot of diversity if it's already created without including niches. The simplest is if I just got one species with closely related subtypes, and I want to ask about possible high-diversity ecological phases. The simplest is what's going to be what happens near to a fitness peak, as when the snowscapes peaks. Okay. So if we look at the closely related strains, so we can look at closely related strains. So these have some fractional abundances, nu I, so I is the set of strains, of S strains there. They have their own phenotypes. They interact with an environment, which is set by the resident populations. And how you characterize each one of these strains is by how the environment affects it and by how it affects the environment. So the sort of the impact vector and there's the effect vector of the environment. Okay. So you can put those together and you can write down what the sort of fitnome looks like. And as a simple form, is this just additive in the simplest approximation on the effects of all the other ones. So you get additive effects coming from the others. And this gives rise to a really simple class of models, for those who've seen before, the Lotka-Volterra models, where the growth rate of a strain has some sort of intrinsic fitness for it, and then how they interact with each other. And they can go extinct, of course, if a nu goes to 0, and I can introduce new strains by mutation. If I'm in this situation where I've got an environment, and they interact via virtual interactions via chemicals, so then this interaction matrix V is just coming from the way they affect the environment and the way the environment affects them. So this is just our simple dynamics for the ecology, the ecological dynamics. And on short times, the simplest is this just goes to a stable fixed point. Some things go extinct, some survive, and form a stable community. Okay. So what does this dynamics of this look like? So here I'm plotting the abundances on a logarithmic scale. So 1's down here, they haven't come in yet. Here is initially start with 1, some other one invades, it comes in, a new mutant comes in there, some things coexist. This red one then is unlucky, another invasion drives it extinct. So on this we have the faster dynamics, which is from the ecology, they reach a fixed point, and then we introduce another mutant. If that's better in that environment, it can come in, but it will later go extinct. So if we look at this on now long time scales, so this is on the evolutionary time scales, where I'm gradually introducing one mutation at a time. Then what happens is all these strains that were in the population, they tend to bounce around for a while as other things are invading and disturbing it. So these environments are constantly getting disturbed by new strains coming in. Then it relaxes to a new equilibrium, then it becomes unstable again. And none of them last very long. They come in at different things, they all last a characteristic time. So you seem to be here getting this continual turnover, only shown a small fraction of the strains, on this slow evolutionary timescale. Okay, and this is the hardest regime to explain the diversification and so on, is when the ecology is relatively fast and the evolution is relatively slow. Okay, and for most of the things that one can imagine with these planktonic things, that's the right regime. A lot of times bacteria, they evolve fast enough that this is not, but then it's actually easier to explain the diversity. It's less of a puzzle. Okay. So what happens if we're near to a snowy peak? So this is now the simplest thing I can possibly write down. I just expand everything inside around the peak. These are just the distances of the phenotype away from the peak. The environment is just set by the average. And I end up now with a very simple function. So the function which is going to give me right, so all of the evolution. Here's strain I. It just has a quadratic peak around the peak. And then it has just a way it interacts with all the other strains. This is just the average across the whole population, and this is how they interact. So I've just got this quadratic thing, and I want to ask what happens there. Okay, very simple. So there are two important parameters. One of these is just the overall magnitude of the ecological feedback. This is the ecological feedback. This is the landscape part, if you like. This is the effects of the snow moving. And then there's the correlation of sort of how much what you do to the environment is bad for you or not so bad. And there's a parameter there, kappa. If kappa is large, it means things you do are always bad for you, like eating stuff or poisoning the environment or other things. Okay, so what happens near to this snowy peak? Okay, so there are a bunch of phases that can happen. One of them is the most obvious one, and that's one where it's almost always clonal. So you look at the number of strains that exist at any time on evolutionary times, and you see this is mostly one, occasionally is two, that coexist, or very occasionally three. And if you look at where these are, they're getting closer and closer to the peak. They're decaying exponentially towards the peak, as you would expect, in just a simple model, so just exponential convergence. Okay, so that's the simplest one. A less obvious one is a phase which is still tend to be not diverse, just a few strains coexisting at a time here, sometimes down to one. But it stays away from the peak. It can evolve forever, continually turning over without slowing down. And in fact, if you start with it nearer the peak, yeah. Yeah, so there's a mixture of, these are simulations, but you can do a lot of analytic things on this model, and you can show that these various phases exist, but you can't figure out the phase boundaries and things analytically. Yeah, so once you're near to a peak, or to a fixed point in this, to a stable fixed point, then you can expand around there, and the generic thing in high dimensions is random matrices that come in with certain statistical properties. Now, it turns out that even if you assume everything is sort of isotropic around there, the behavior is pretty similar. So you can make it even simpler and do more analytic things. Okay. But we can show that a lot of that's robust, and then these phases all exist for the more general models. But it's just basically this simple quadratic model here. Yeah, so this basically simple model is just quadratic. It's as simple as possible. And then, say, the U and A and B have some statistical properties as random matrices. But if you said you want to just make those proportional identities, the same phenomena occurs. Okay? With some extra simplifications and complications. Other questions? We can't just have XBEL Labs people interrupting, even though there are various other XBEL Labs people here. Complexities. Right. Right. Explores that. Right. Yeah, so yeah, you can use, so the field of high dimensional random things is now a lot of activity in it because of machine learning things and so on, earlier from neuroscience and other things. And so you can think of these things in various ways, and the sort of analytic ways one does it are actually related to some of the methods that are used in other areas.
So one thing I've been wondering are the differences among the cyanobacteria that you were discussing at the beginning. Are those larger differences than differences we see here among the people?
Yeah, but your genomes will typically differ by 1 in every 1,000 bases along the genome, and this particular species, they can differ by 10%. Okay, yeah.
So they are.
Yeah, it's unusually diverse for a bacterial species. Bacterial species are a rather arbitrary sense, but I'll say right at the end as to why this is actually sensible in this case.
What kind of approximation would you need to get that mean?
Well, this is Darwinism. I mean, Darwin is just an evolutionary process which is very general, right? So in some ways so general that it's very easy to make models and do simulations and produce pretty pictures and get them on the cover of glossy magazines. but learning stuff from him is much harder, right? But this is all, it's all Darwinian. So that may be some of the general questions I can come back to at the end. Okay, so we were talking about the phases. So this phase now doesn't approach the peak, but it's not diverse either. But then I get a phase here which gets much more diverse, and here the diversity gets to be of order the sort of dimension of the environment. That's the maximum I get. But nevertheless, it still converges towards the peak. And then the most interesting one, which is the diverse red queen phase, where it gets continually turned over. You see it diversifies here. The diversity, again, is proportional to the dimension of the environment, but it never approaches the peak. This flattens out at long times here. So this is the kind of phase that we were sort of, hoping for, that you get something which is diverse, continues evolving forever in this red queen way, but is not now a single population, it's continually turning over populations. And so this is rather robust. It can occur in lots of circumstances. But of course, we don't know in any realistic model, we don't know whether this is what occurs. Okay, so you can put these all together to get a phase diagram, and there's actually another phase and things that can occur as well. The crucial thing to note here, physicists tend to like having a Lyapunov function. They like when you've got some function that you can maximize. Ecologists also like such models, and biologists in central New Jersey like such models. However, they are very special. Once you've got Lyapunov function, you're always going to converge towards the peak. As soon as you go away from that, you get the possibility of things being different, right? So little bits of non-gradient ascent can really make a big difference. Unfortunately, we're now stuck with not a lot of diversity. Very generally, it was a long time ago that interactions via some number of chemicals, the maximum stable community size you can get is the number of chemicals, and in most sort of models you tend to get quite a bit less than that, and that's to do with the rank of the interaction matrix. So one question is, can stable communities continually evolve with much higher diversity? Ideally, one would like something S maybe which is exponential in the number of chemicals. If you say you've got chemicals that you get carbon from, ones you get nitrogen from, one you get phosphite from, I'd at least like to multiply those numbers of different sources instead of just adding them. But there's another interesting possibility, is that maybe I shouldn't be looking for stable communities. Nothing says that I can't have unstable communities. They don't stably coexist. We can also ask what different aspects then, other than how you affect the chemical environment, what about other ways? And again, how much complexity is enough? Okay. So the sort of next simplest thing one can look at, and this is really saying, okay, what about pathogens? So is to look at a bacterial species, which are all identical, and phages, which are all identical, except for how they interact with each other. So the only difference between them is how they interact. Right, so this, to the first approximation, they're all people identical, but you have but how you interact with the COVID strains is different because your immune system is different from each other. But otherwise, you're identical. So this would be like that. And then there's just generalizations of the Lotka-Volterra interactions that you get for here. So we can ask, what does this do? Okay, how diverse can this be, or what are the dynamics? Okay, well, there's some very simple dynamics which occur when you get interacting pathogens and the hosts, is what's sometimes called kill the winner or escape the killer. So we've got here a bunch of bacterial strains in blue that are doing well, they're coming up. But then a phage that can attack those ones, they will tend to come up. But that will, of course, suppress these blue ones back down again. But then some other bacterial strains, which are more resistant to this phage, they'll come up and take over. So you can get this continually going up and down with the continual blooms and busts. So something like this shows, geez, okay, this could keep on going and maybe be a way to get a lot of diversity. Okay. Well, and here's nice for statistical physicists. This is a sort of partially exactly solvable model where you can show what happens. You get a persistent chaotic state like a state like this. Unfortunately, as with a lot of exactly solvable models, they're not robust to adding features. So what happens, if I go away from that, you get this situation where initially you get this chaos, but then the fluctuations get wilder and wilder and wilder, and eventually, even if your population was 10 to the 27, you will go extinct. Some of you will go extinct, and you end up with a small number of strains left at the end. Okay? So it goes unstable. The hosts and pathogens by themselves can sustain diversity for a bit, but it'll drive extinctions, and you lose that diversity. So we'd like to ask, can this be rescued? Can the community be rescued and recover from extinction? Right. So this is, of course, something which the ecologists have thought about. And this is particular whole field of ecology called island biogeography, where you think in terms of an island, and here's where you're making all your observations. And then you have the mainland, and the island is not very complicated and not that big. Species can go extinct here, but then they can get repopulated from the island, from the mainland. So there's a lot of things done on what the statistical properties of what you see here as function of the mainland, and there are experiments and so on. The problem is you have to assume that there are all these strains that coexist on the mainland. So this just pushes the problem and is rather a cheat as far as sustaining a lot of diversity. Okay? So what we'd like to ask, can you do without that, but using the general idea that you have some spatial structure? So particularly we would like to have, here's our caricature ocean, we'd like to have mixing around with the ocean or migration of the motion of the organisms, with no spatial gradient. So I would like to ask if it's completely uniform, but things are mixing around. Some strains then can go globally extinct. That certainly is going to happen. But what we're going to ask is, can many strains still survive? Can I get a lot of coexistence in this situation? And some work of ours, and also Roy and collaborators on a somewhat different class of model, showing that you can get chaotic survival in this kind of phase. So what happens is that you get a spatio-temporally chaotic phase where the dynamics locally on each location is chaotic, but it goes out of sync in different regions. So if it goes out of sync, it means that if you're doing well in some place and you're then dying out, but your relatives are doing well somewhere else, they can repopulate where you are coming from the other location. So some of the strains go globally extinct. All the surviving ones have this blooms and busts. You can't survive unless sometimes you have a bloom and a bust. But the crucial thing here is this phase is very stable. It survives for exponentially, sorry, this is an older slide. It survives exponentially long in the spatial extent. Okay, so in the spatial side of the system, survive exponentially long. So we're sort of turning modest timescales into very long ones by sticking them up in an exponential, which is one of the goals. So what does this look like if you look at it at one location? So here you're seeing the bacterial phage bouncing up and down. We're only showing a few of them. There are many more that coexist. You can understand this by complicated methods. Coming back to a question there for theorists, I can say something at the end if people are interested. Okay. So what this gives rise to is a spatio-temporally chaotic phase, which can sustain high diversity. I can have a lot of strains which coexist in this. And the fluctuations are wild. They're on a logarithmic scale. Okay. So what I'm showing here is a space-time plot. So here's a one-dimensional system. There's space. Here's the time. And this is on a logarithmic scale. And this is showing one strain. And it's showing one strain which is wildly going up and down. And you see these whites are where it goes locally extinct. But then it's getting repopulated. And you sort of get these waves of repopulation and waves of the effects of the extinction. And this has a very interesting statistical dynamics that we only partially understand. If you look at one strain looks like it's in some spatially-temporally varying environment set by its interaction with all the others. So if you're a phage, there's the statistical effects of all the bacteria that you're interacting with, in which you're interacting differently with each of those. And that statistical effect gives you like a fluctuating environment. However, that fluctuating environment is tuned by the dynamics just to give this kinds of behavior. If you put a sort of generic fluctuating environment, you wouldn't get this. You would tend to drive extinctions, or things would just stabilize. So this is a sort of self-consistent way of the interaction between the bacteria and phage that gives you the spatio-temporally varying environment. And this we have quite a lot of understanding, not a full one of it, but this seems to be a very robust phase. Yeah. So there are sort of wavy things here. There's some intermediate regime where the spatial spread has an exponent of 5/6, we think. But there is definitely spread. This is because of the migration, because of the mixing. So if I've got something which is low here, it can get filled in by something which is going on then. And that'll be growing because the correlations in the environment, and that'll give you these structures. So I think we have some understanding of this, but it's still relatively limited. Okay. So now we come back to the question, how much complexity do I need to get this? Here I just put in interactions between the bacteria and the phage by hand. I didn't say anything more than that. I could have arbitrarily large number of strains in there interacting with each other by God knows how. Okay? So I want to now put in some direct way they can interact. So a typical way that a phage interacts with a bacteria is the phage has a tail, and the tail binds to some receptor, a particular protein on the surface of the bacteria, or in case of COVID, on a particular bacteria a particular protein in your cells. And this then will depend upon both, of course, the nature of the tail and the nature of the receptor. Okay? So if we say these are d-dimensional vectors, so d is going to be some modest dimension, so the number of amino acids, say, that make up these receptor in the tail. And then the interaction is going to via the binding energy, which, simple physicists, we just take the dot product of these two vectors. Okay? So this is the binding energy, which depends on the strain of bacteria and the strain of the phage. So now I've got phenotypes. Well, there's the phenotype of the bacteria is its receptor, the phenotype of the phage is its tail, and the interactions are between those. So I'm not just cheating and saying I've got random interactions, I'm putting them in here. Okay. And then we can ask what happens. Well, we say that the way that they die, that the bacteria will die from the phage, and the way the phages grow from the bacteria is just some function of this interaction energy. And then you find that there's a chaotic phase that can come in with these phages. They're generalists. They can each interact with many other bacteria. However, if you do the simplest thing, you just say this is linear, you again go back to this boring result that the maximum diversity you can get is of order d. Okay, so of order the size of these vectors. However, as soon as you go to a nonlinear function, just sigmoidal function, you can get much higher diversity. Its dimensions bigger than about 4. If you look at how they interact with each other, you see the reds, the blacks, and whites. There's no particular pattern. These ones went extinct. These ones survived. This is a bit analogous to memories in Hopfield models. So the original Hopfield models did not have very many high memory storage. People have worked out recently how you can sort of get exponentially more storage with kind of nonlinear interactions, which are just of this sort of kind. You take a low-rank matrix and you take a nonlinear function of it. So this is very analogous to that. Here we only understand it, I say, in a very partial way. Okay. But nevertheless, this shows us that we can get high ecological diversity from simple biology, just these simple interactions, without assuming anything about niches. There's nothing niche-like about these interactions. Yeah, it's not the eigenvalues. It's much more complicated properties of the matrices. Yeah, because all these things are very nonlinear and so on. And so you can do some things. I can explain more. Yeah, after. Okay. So now, of course, we want to combine. We want to combine the evolution and ecology, and we want to ask, can it diversify? Can it continue? What limits it, and so on. So we want to look at the evolution of this phase, because this is the one. I think really sometimes the only example that's known of a phase that can occur in simple models without putting in a lot of each one has its own niche. Okay. So I want to ask what happens as we evolve it. We want to again evolve things slowly. So the evolution is the tail changes of the phage, or the receptor changes of the bacteria, so it changes their interactions. We want to ask, does the diversity increase? Does it saturate? Does it crash? And the answer is all of the above. They can all occur. We can get different regimes. We partially understand this, but only very partially. This is still ongoing work. Here's an example. Here it's now low dimension, just 8-dimensional receptors and tails. You see that the diversity is going up, is continuing to increase. We don't know how far this is going to go. This we're now looking at it, you can already with somewhat lower dimension get a lot here. What quite happens as we go along here, we only partially know, but the conjecture is that it's possible that you can get exponentially many phases in the dimension. It's not going to keep on going indefinitely, but if you can get exponentially many with this very low complexity of just these interaction with the tail and the receptor, This is something which one is sort of looking for. Somehow bootstrapping small amount of complexity as far as how the organisms interact with each other into giving you a lot of coexisting diversity, which can continue evolving in a red queen way. Okay. So we've talked about the eco-evo dynamics in high dimensions, how in more dimensions the behavior is definitely different. Simple models give you different kinds of phases. The continual evolution diversification is less surprising, at least less surprising than it was to me initially. We at least have, in a very loose sense, some kinds of plausible scenario. Okay? Particularly with the bacteriophage, we have this robust spatiotemporally chaotic phase with extensive diversity. But now we want to come back to our little cyanobacteria, and we want to ask, is the diversity of strains the right question? Okay, here I was just doing evolving populations with mutations and so on, but there wasn't any sex. Okay. So for Prochlorococcus, is the diversity genomic diversity or is it genic diversity? What do I mean? If I look at some parts of the genomes, so I'm going to look at a part of the genome here, and I'm going to look at a single gene, and I'm going to then draw a family tree of all the variants of that gene in the across the population of all these cells that I've got. And I'll draw a little family tree here, and I've drawn a simpler one, so on sort of the larger scales it divides into two groups, but all kinds of substructure. Then I look at a gene somewhere else in the genome, and I again see I get some kind of tree. Again, it has this structure of two bigger groups. And then I try to ask which of these match up with which of those. And you find it's a complete hodgepodge. And so if I just do schematically, just in terms of these two types, I get here, I've ordered these, so little a's and little a's. These are the different genomes, but the little b's are randomly linked with the different a's. And what's found is that there are very little correlations along the genome. Okay? This is something which cannot occur if I have asexually evolving populations just evolving by mutation. This can only occur if I have things that move around bits of genes between genomes. So bacteria were long thought to be primarily asexual. Then people started realizing there was gene exchange. In recent years, it's become clear they have a lot of gene exchange. Prochlorococcus was supposed to be an example of very asexual, barely any genetic exchange. Nevertheless, on the large scales along the genome and on time scales associated with this whole species, there is a huge amount of recombination, and these correlations are broken up going along the genome. Okay? There's little ecological or geographical correlates with the variations of the genes. So this we had dubbed in a different context. We first realized the extent of this, a quasi-sexual bacterial species, that you can think of each genome as sort of been a random mixture of variants of each gene. So that's roughly speaking what your genomes will look like. There's a lot of sex goes on in humans. Some bacterial species, another cyanobacterium from Yellowstone Hot Springs, by some dimensionless measures, it's more sexual as genomes than humans are. Okay, and if someone's interested, I can explain what I mean by that. Okay. Then we now have to get to the question of what sustains the diversity of each gene. We don't know. But the simple question we can ask is what evolutionary process could give rise to this? Right? So now we're not talking about understanding the ecology. We just want to ask evolutionary processes that exchange genes around to give to this. Okay? So what this process is you can hitchhike genetic diversity on segments of the genome which are good. So these could be a segment which is good because it makes you resistant to some phage which is common where you're living. So here's now, say, a mutation or some other change on the genome which is good. And then there are a bunch of mutations which these genomes differ by, which are just sort of markers. They're neutral. They don't do anything. Particularly, they don't change amino acids. So you have these bunch of these marker genes. And then we have a gene transfer. So this then segment gets transferred another genome. And then that's now good. So this genome with this now on it can expand. So I get more copies of that genome. Some of the other ones are around as well. That's the selection acting on it. Then I can again get this happening, and I can get this happening here. I've now moved around things well. I've dragged the blues with me, but not the greens anymore, and eventually got into this, where the reds in this simple model take over, because they're the good things, but they've hitchhiked with them. They've hitchhiked along some of these blues and not as many of the greens. Okay? So what this can do, it can change around the abundance of these blues and greens even though all they're just doing is keeping track. They're just markers, right? But the nice thing is from DNA sequencing is you can use those as codes and you can follow those and you can look at what the consequences of this for the statistical properties of the genome. Okay, so the thing you usually can measure is just snapshots of a population and measure the statistical properties of the genome, and you can hope, as one does with a scattering experiment or anything else, that you could infer something about the dynamics coming from there. Okay, so what are the statistical properties? So the last thing I just want to show is one very classic property that population geneticists love, and that's to look at these synonymous mutations, ones that don't change amino acids, and you look at the site frequency spectrum of those. Okay, frequency is used in a different sense than biologists. It means a fraction. So you look at sites along the genomes, and you look at whether they have, say, a G or a C or an A or a T. We're just lumping those together for simplicity to make it like an Ising-like variable. So here's now looking along these genomes. On this site, some have A's, some have G's. Looking along the genome, some have C's and some have T's there. And so we can look on this site, 60% of them have a G or a C. On this site, only 30% of them have a G or C. And then we can look across the whole genome at all of these sites, and we can ask what are the statistical properties. And you get this. This is showing as a fraction of sites from 0 to 100%, which ones are there. So over here is almost all ATs. Over there is a GC. And the data here are shown in the blue. And then I've shown 3 theoretical curves here. The 3 theoretical curves, one is the classic one, which the population geneticists love, which is just stochastic birth-death or effective birth-death processes. That's this black curve. Then there's a curve which has been suggested, just a sort of uniformly expanding population. And then there's this red curve. Okay? Now, all of these curves just have 2 parameters. Basically, one of the parameters is just the asymmetry here, and the other parameter is essentially the variance, which is associated with times to common ancestors. So they're the same 2 parameters that are just computed directly from the data. This red curve here is this picture I showed of this sort of quasi-sexual hitchhiking on continual selection across the genome. Okay? It is embarrassingly good close agreement with the data. I don't understand why. I think it's somewhat fortuitous, probably, but it's certainly a lot better than these and clearly captures something. And the thing it captures is that some of these sites, so if you're a little blue circle on one of the curves that I show, some of you occasionally get lucky, something happens on the genome which you're on, and you then get expanded way up because of the things which you're linked to along the genome, the good things that are going on as they're moving around. So you can only get this kind of curve with that sort of kind of big effect hitchhiking. So it's very rare in population biology or evolution that you ever get a curve that looks as good as that. And I say I don't quite understand why it should be so good. Can you? I just have one more last slide, and then the questions. Okay. So we want to end with more questions, as Oscar said. I like the questions. So now, instead of saying why is there so much genomic diversity, we have to say why is there so much diversity of each gene or each combination, a group of genes. Okay, and here's where we have direct evidence about my statement I made very early on in my assumptions that everything was conditionally beneficial. Right, so supposing something had come in the prokaryotic genome that was good for the whole population, then it would have just taken over, and I should see some place in the genome where everything was more closely related than other parts. Okay? And you don't see that. Nothing is taken over of the whole genome in the last million years or so. So it means that everything is only good sometimes. It could, of course, be good as long as you're not in the majority. If you get in the majority, then it's bad. Okay, and that's particularly the being attacked instinct by phages is going to give things like that. So this is sort of the starting point is supporting that. And Prochlorococcus is incredibly good at doing things. Right? If you say there's a good change, a beneficial change that involves this particular mutation here, this particular mutation here, and this particular mutation here. Okay? So that has a probability of about 10 to the minus 27 happening in one generation. If you say all the combinations are bad unless you get exactly those 3, all the intermediates are bad, that still happens every day because the population is so huge. Right? So it's really good at producing genetic variation. It's not known how genes move around. But they can move around very rarely. But if they move around and they're good, then they can rise to big population. Right? So all of the action going on with evolution is changes that happen in one individual, but then they can come over and take over the population, and of course you don't see them. Right? So this is why so many genetic variants were discovered in South Africa. Okay, Oscar can explain that. Okay, so one of the questions here, we sort of have this loose idea that somehow bacteria and phage things can do it, and maybe all of this ubiquitous moving things around the genome is actually associated with the phages and phages moving things around. It's not clear that that will do it or what the other possibilities are. Okay. But we can also go back and ask, geez, here was a way with pathogens that we can get extensive diversity from relatively simple genetic differences. Can we also get it via chemicals by some kind of nonlinear interactions like this? Or is it that, in fact, host-pathogen interactions are crucial for generating and sustaining diversity? Right? Do you need to have things trying to kill each other, or can you just, by competition, get that? And that we don't know. And then the sort of overall question, how much complexity does one need? How much does one need to be able to explain? Do you have to have all the details of biological complexity? Or maybe there are relatively few things in the way that organisms interact with each other and with the environment that's enough. Okay, so I'll leave you there.
Further questions? In the back.
Yeah, thank you for a great talk. I'm wondering how seriously I should take you when you say there are different phases that you discovered. Do you literally mean order of parameter,
Yeah.
like one of the universality classes, or just like different phases?
No, I do in that sense that there are qualitatively differences which can have quantitative measurements of them, analogous to order parameters, and these phases are robust. You can change things around in the models, you can change parameters, and they have a range over which they're robust. Okay, so they're in that sense. Now, then you can argue, well, why do I think that biology is in the interesting phases rather than the uninteresting ones? So if I come back to my original one of the fitness landscapes, some fitness landscapes just have a small number of peaks and you get to one of them. Okay? But if you get to one of them and then the conditions change, it's really hard to get out of it. If you have this continually moving around, and with all these settles, and even more if you have a diverse population, you can much more easily adjust to when conditions change. Right? So if I ask the crucial bit about evolution, which is that everything we see is conditional upon a very anomalous evolutionary success, right? Sometimes on short time scales, like pathogens, sometimes over billions of years time scales. So everything conditional on success, you can say, well, that's likely to have properties that make it more evolvable, even though there wasn't anything that drove it that way, but just the condition on us seeing it. So I would say that these being in this sort of snowscape phase is much more evolvable, so the populations that we see are likely to be in that. Okay? So you should take it semi-serious, I would say, in a sort of suggestive way, and really what we're trying to do is develop scenarios, right, rather than some explanation of a particular set of data or experiments.
So in the models you've used to describe the interaction between the strains of, for example, the bacteria and the phage, is there a pathway to constrain what kind of interaction will like impose some properties on these random matrices that would provide some physical understanding about what is going on, or is it different?
Yeah, I mean the nice thing with some random matrices things, there's certain correlations that you sort of need that can determine things, and beyond that, not so much details matter. Now, of course, it's not really random matrices and so on, but a lot of things making up from sums and differences of a few things, and then you take a nonlinear function of that, it behaves a lot like a random matrix as well. Right? By the time you get to 5 dimensions, infinity is a better approximation than 2 dimensions was. Right? So I think a lot of it is their sort of experience from other contexts where you don't need that much complexity to be better characterized by some kinds of randomness. But then be careful about what the statistical properties are that you're assuming.
So you started with this thing that kind of tells me about the time scale separation between intelligent and
Yeah.
Does that still hold in this picture of getting exchange and then phages? I mean, is that all
Yeah.
at the same time?
It could be happening on the same time scale, and there'll certainly be a mixture of time scales, but it's not clear. There's also the migration time scales, how much they're mixing. What we know is that surface oceans can mix, and in this case around South Africa, in tens of years. We know that genetically, that you can find a very close relative between the Atlantic and Pacific that are separated, but not until separated by tens of thousands of years. But you can find a gene variant in the Atlantic and Pacific that are much closer relatives. Okay? So it may well be that the sort of genes can move around between oceans much faster than the individual bacteria. Now, of course, we don't know that if you sampled, 1 million cells from the Pacific and millions cells from the Atlantic, you may well find ones that are much more closely related. So the balance between the sort of mixing or migration timescales and the evolutionary timescales and the ecological timescales is very difficult to know and understand. Most species, one doesn't have information about the spatial mixing. Human pathogens we do, right? Oscar's done very interesting things on this using information from genome sequencing to try to infer stuff about. Most bacterial species we don't know, including ones in our guts. We have a sense, but we don't know that well. The nice thing in the ocean is we have a bit more measure of it, but you can't really measure the things you want, because doing a tracer in the ocean, the tracer can't, if you put a chemical tracer on it, you put a lot of rubber ducks in the ocean, and you watch them going around, it tells you things, but the evolutionary process means that a tiny bit that gets somewhere, and it happens to be good when it gets there, will take over. So you can't detect that from tracer kind of measurements. Right? So it's really hard to get at the kind of sort of potentially very rare events that are relevant. And it's a similar thing with the evolution, right? Does it matter for the evolution of Prococcus that it's 10 to the 27 cells? Or could it be that, it would evolve pretty similar if there was 10 to the 18? Right? And we don't know.
In the back there?
I was just saying you can get some set of genes that control variability of the gene.
Yeah.
That gives you sufficient variability.
Yeah, so there's ways of getting, and particularly in more complicated organisms, that for a given genome, their properties can vary a lot. Right? And so you can get the cells in your body, they all have the same genome, or almost all of them, but they have very different properties. And so even bacteria can do some amount of that. Yeah. And there are also genes that can affect how high the mutation rates are, or how easily you can exchange DNA with someone else. And so all of those things are complications on top of it. I would say generally all of the complications make it easier to get more diversity. Right? And so the fact that one can sort of already see it in pretty simple things is saying, okay, there's things one would like to understand more, but that one doesn't need all the complexities.
All right. Maybe we can take do you want?
Yeah, I'm happy to. People can leave. I'm happy to.
Okay, so let's
Stay if
keep
people have
going, asking questions, but people need
People,
to...
yeah,
feedback, please.
yeah.
So I was actually wondering that you brought in nature in the Snowscapes.
Right.
Because of that, would there be transient phases that you have missed in your phase time?
Yeah, no, there's definitely transients, and actually even around this peak, there's quite long transients that can occur. In particular, there's one where it occurs where you get closer and closer to the peak, and then the population splits and you go shooting off and it goes unstable. And that's actually one that was known from before, sort of branching phase, where you think you're stable, but you're actually not. And so yeah, there are definitely transients, and this is a sort of mixture of simulations and analytic things and sort of heuristics and so on, and it's at that stage. There are a few things that we can really work through, push through analytically enough to sort of know that we're solid, that we're not fooling ourselves by having dimensions of 20, of seeing something that looks as if it's there, because we have enough understanding of the sort of high dimensional regimes to at least know that we're not fooling ourselves in the simulations. Uh-huh. Oh, okay. That's yeah. So there's what we believe is that there is a strong asymmetry in the mutation rate. That the mutation rate in this direction is a quarter of the mutation rate in that direction. And that's where this asymmetry comes from. Now, there's a possibility that there's a different source of the asymmetry, but at least at this point, we haven't tested that as well as we should have. So mutations can go from, say, a C to a T or a T to a C, right? And one of them is different than the other. One rate's different than the other. And so typically those rates are somewhat different. In this particular organism, they're very different, it seems.
Do you have any more knobs on that? That we
So the theory, there's no more knobs. If you could, there's a natural knob to try to put on, which maybe improves the fit down here a bit better. We would really like to have, this is, the sample depth of how I had to do this is only about 120 cells. We would really like to be able to do it to 1,000, and then you start seeing extra features in there. And I'd be extremely surprised if this works as well there. You see there's a little bit of an extra peak there, which is sort of a natural thing that one would be surprised if it wasn't there, but it's remarkably tiny. So, yeah. Yeah, I know. It's embarrassing. Yeah, I don't take it too seriously, but it's a nice figure to be able to show at the end of a talk, which is all hand waving. You know, some data. But okay, well, thank you.